Okay, guys. Um, so I think I let too many people into this class by mistake, and so there might be a chair issue sometimes. But at the same time, half the people who I told were in the class never got back to me about an ad slip. So maybe I didn't let too many people in. We'll see. Someone might have to stand. There's a chair in the corner. Anyways, if anyone still needs to add this class, the deadline was yesterday, hence the email that went out to everyone. Did you get that? That said, see me today, which was yesterday. I'm sure you can still add it, but Dolores will give you and me evil eyes. You guys all know Dolores, right? Yeah. So she's your lady. She'll give you evil eyes and just blame me because I'm new, kind of, not really anymore, and I don't know what I'm doing. Dr. Ehrlich, she has no idea what she's doing. She told us to come on Wednesday. Anyways, but that actually highlights a very important message I would like to pass on to you. Check your email frequently. Who checks their email once a day? Who checks their email every 10 minutes? Who doesn't really check their Towson email? They only do Facebook. Okay, because I've had that problem. So make sure, it says in the syllabus, make sure that whatever mode of communication that is not face-to-face that you use, or phone, that you use, that it gets the Blackboard emails, because I might email you via Blackboard about something important. I might have emailed everyone yesterday morning saying there's a pop quiz right now. How many of you guys would have known about it? Okay, good. At least half of you. So that's good. So just be on it. I am obsessive and check my email like every 2 seconds. And that's not healthy. But you probably want to do like at least, every 2 hours. I don't know. Maybe a morning and a evening check, perhaps. Anyways, the point is I want to communicate with you guys, especially when you're begging me to let you in the class and I say you're in and I hear nothing and no one comes to get ad slips. That tells me that you guys are not checking your email. Okay, enough of that. A word about the pretest. The average was 6 out of 10, which was really scary. Yes, I corrected the one that was wrong. So everyone who got that right got the point for it. The really scary thing is half the people that got that wrong truly got that wrong. Now, a really disturbing thing was the number of people who took the test again, wrote all the answers down, checked them all off, and gave me the wrong answer for that one that was wrong, right? So someone changed a perfectly right answer that was corrected wrong to the wrong answer in an attempt to cheat. So that really that angers me because A, it makes me think you think I'm dumb, which, I mean, I can't be dumb, but I'm not when it comes to the quiz I write. And it makes me not trust you guys. And I like to start off trusting the class. And it wasn't one person, it wasn't 2 people, it wasn't even 3 people. It was closer to about 10 people who closed the quiz and then took it again with all the right answers. So I can tell how many times you take it. I can tell how long you have it open. I can tell everything. I'm telling you now, work with your friends, work with your classmates, look in your book. That's not cheating. Cheating to me is going A, B, A, B, A, B. What are you learning? Nothing, right? So what's the point of being in a 400 level class if you don't want to learn anything? So I'm telling you now, because A, it's a waste of your time because your first attempt is what counts. So those of you who thought you got a 10, who really got a 3, you got a 3. And that's part of the reason that the average is low, probably because of the people who thought that they could game the system. So stop it. Darn it. Okay. Yes?
I wasn't able to see, on the pretest, I could see the ones that I got wrong, but on the quiz that I took, I couldn't see them.
Oh, really?
Yeah, for some reason.
Okay, I may have just not checked the right box.
Okay.
So I'll double check that, and I'll make sure to have that checked in the future. So the question was on the quiz this week, people couldn't see what they got wrong. I apologize because I could have forgotten to check a box. Yeah. Yes.
How difficult to find in the book online?
Okay. Okay, we can do, I'll stop 5 minutes early and show everyone how to find the book online on the big screen. So we can go through that. Yes?
So will we eventually be able to see the answers to the past quiz?
Yes, I will make them available somehow. Email me to remind me. Okay. Yeah, so repeated attempts are obvious. Okay, so that's enough of me screaming at you guys. Sorry, I'm a mom. I've got 3 kids, and I'm used to like. So yeah.
So people who are on the waiting list.
Yes. So people who just got into the class, please let me know when you have access to Blackboard. I know who you guys are. Some of you already are on my Blackboard. Some of you still have to be added. Once everyone's on, I will give you guys, 3 or about 6 days actually to complete all of those. Okay. So don't worry, you guys are not losing out by being late. Okay, so based on the pretest, I wanted to clarify a few things. There were some things that were really disturbing to me. Okay, so this was the one that was the oops on my part. The genetic code refers to these answers. So who wants to tell me what the true and correct answer was? C. Right. The triplet code in the RNA that determines the amino acid to be added by the ribosome to the growing polypeptide chain. I got a whole lot of the bases that encode the genome. It's if you Google genetic code, pictures of the codons come up. So I want you to realize that it's a code in the sense that it contains information. Those 3 bases contain the information about what amino acid is going to be added. That's the code. If you have a code, you're decoding something, right? So just having bases, that's not a code. That's just bases. It's that triplet code that encodes the amino acid that's going to be added. I got a lot of the amino acid sequence that makes up a protein. No, it is not. So if anyone has questions about this, I would love you guys to come to my office hours or email me, because this is very basic information that you should know coming into this class. Okay, so this one got a lot of people. And that's good, because I need to know where you guys are at. So the identity of the following protein is likely to be. So who thinks they can tell just by looking at it?
No, I can't tell, but I have a question.
Okay. Okay, so you don't have BLAST in your brain?
No, I don't have BLAST, but I put it in I went on GenBank and I put it in the search bar by protein, and I didn't know we had to go through BLAST. So.
Okay, well, a lot of this is for me to know where you guys are in things. Okay, so the way this works is, let me just come out of this for a second, and let's open up and do it right here, right now. Oops. Okay, so if you go here, you're going to go, oops. Okay, so you go to BLAST, right here. If you guys have never seen this before, please let me know. So it's a protein, we go to protein BLAST. So this is how people identify proteins. Let's say you're working on something you don't know anything about. You're a researcher and you are studying, let me highlight this. Can't do 2 things at once. Okay, so I put it in the box, right? There's your sequence. BLAST. We'll see what comes up. So let's say you're a researcher studying a protein. And a common way to identify what a protein does is you're going to see what it interacts with, right? So you actually pull down your protein. Whoa, look at that. And so that's giving you some conserved domains. It'll come up in a minute. But already it kind of tells you. So you'll pull down whatever protein and you'll look for what proteins interact and you'll send them off to be identified by mass spec, and that just is a way of identifying proteins, which you'll go through maybe at some point. But what they give you back is sometimes just little peptides. And what they do is they blast them, and based on homology, right, similarity to other proteins, you usually can ascribe some kind of function to your protein. So this is a really useful thing to know and understand. So if I give you a random sequence, you should be able to identify it like that, right? Okay, let's see. Well, right here, it's even saying actin right there. Without even the results coming up, it comes up as actin superfamily. Eventually, there we go. Oh, actin is highly conserved. Look at all that red. Okay, actin, 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 actin. What do you think the answer was? Actin, actin. Okay, so that's how you do that. My apologies if people didn't know. My apologies if people didn't know the single letter code. But this is a pre-test because it's things that I believe you should know. And now I know you didn't know, so I can teach it to you. Okay, so let's go back. So that was that. So it seemed like a lot of people just randomly guessed. So now you know. What are you going to do? Okay, next one. Gene expression refers to, this one confused a whole lot of people. And then I went to talk to Dr. Bulmer about it to make sure my question was fair. So what do you guys think the answer is? I heard someone say C. Who thinks it's C? Raise your hand. Translation, okay. Anyone have another guess? B, yeah. So translation of a protein is not gene expression. The true meaning would be the whole flow of information. But when you talk about gene expression and regulation of gene expression, you're talking about what actually determines if a gene will go from DNA to RNA, so transcription, right? And then things have to be translated, and then you express the protein. But transcription is the best answer here, right? And so the reason I put this in is I want to know if people understand that gene expression occurs in response to some kind of stimulus, some kind of, it doesn't just randomly happen, there is some kind of environmental cue or something that causes a gene to be expressed. Any questions about that one? Okay. Okay. This is another one that I think confused a whole lot of people. I'd say about half the class got it right. Translate the following. So ATG right off the bat is a methionine. So then there was really only one answer. You didn't even have to truly translate it, right? But I got a lot of D for some reason. I'm not sure why. But so have you guys seen this kind of, have you guys seen this kind of thing before? This, the genetic code, right? So ATG, which would be U in RNA, G, methionine. So it's like a clock. So anyone can Google genetic code and get this in Google Images, or I think it's in the book, maybe in the front or the back. So that's how you would have gone about doing this one. Did anyone have any questions or want to explain to me what was confusing about this question? No.
I have a question.
Yeah.
So, trying to solve this, you said, I guess it would use using the letter code and not necessarily that. Okay, so you said the answer is C, right? ATG said it's methionine, so that's M.
Uh-huh.
And the next one, GAA.
Uh-huh. So then you would go, so let's say you want to really solve it, right? G, A, A, and which is E.
G.
Which is E.
How are you supposed to know G goes to E?
Oh, so you guys really do not know the letter code. Okay, well, you look it up. That's one thing to do. This wasn't a test, right? It wasn't like sit in your, you always can look up, use Google, use whatever you need to get to this, right? So yeah, so maybe I chose the wrong one, but you can get the same chart with the letter code if you want. Yeah. But either way, the dead giveaway was ATG being M because it was only one option. So, okay, so definitely I want everyone today to go home, go on your computer, Google genetic code, and find one that has the abbreviated code and the letter code, and just have it on your computer in a file somewhere, and eventually you'll know them all. You don't need to know them all. I'm not telling you to memorize them, but it's useful to know that they exist and to know where to reference a chart somewhere. I don't know them all. Questions? Okay. Okay, that's enough about the quiz. I'll do this again with the weekend quiz for a few minutes on Friday. Okay, so we're going to start with Unit 1 on intracellular traffic. What do I mean by intracellular traffic? Trafficking of what? What do you think? What do you think I'm talking about trafficking inside intracellular, within the cell? Trafficking what? Trafficking cytoplasm? Proteins, right. So we're going to talk a lot about proteins and how they get to where they need to go so they can do what they need to do. But I'm going to start with a tiny bit of review because I'll feel bad if I don't. About basically how do you get to the protein. You guys should all know this, but we'll just go through it for fun, right? So we'll start with talking about going from DNA to a functional protein, which would be like, I guess, gene expression, right? Okay, so it begins with transcription, and then you get translation, right? So you've got your DNA, and then your RNA polymerase binds wherever your promoter is and makes RNA, right? Your RNA is matured, comes out of the nucleus, and then binds to the ribosome, and then you've got your polypeptide train, right? So that's gene expression as a whole. But pretty much, this is how you get your protein, right? And then your protein comes out of the nucleus, and then what? What has to happen? What did you say?
Go where it needs to go.
Go where it needs to go. Yeah, go where it needs to go. Right. Okay, so what do I mean, go where it needs to go? Like, don't proteins just hang out in the cytoplasm? No. So, right, so we talked about this last week a little bit, right? So every protein has a job and has a place where it functions. Another very common way to identify the function of a protein, right? Because not all proteins are known what they do, especially if you work on understudied organisms like Plasmodium, right? What does Plasmodium cause? Malaria. I heard someone say malaria. Actually, there's so much money being thrown at Plasmodium, it's not even funny. But still, there's so much not known. But so one very common way to look for a function of a protein is look where it is. Do microscopy, get an antibody to some, your protein and some organelles, and see where it goes. Does it go to the ER? Maybe it's part of the secretory pathway. Well, anyways, does it go to the plasma membrane? Does it go out of the cell? What does it do? Where does it go? Because they function in certain places, right? Okay. This I had to put because it's obligatory, but you guys all know proteins fold. And if they don't fold, do they work well? No. Okay. And they need to get to the right destination, right? That's the whole message here. Okay. So gene expression is regulated by external signals. I keep talking about some kind of stimulus, something from the outside, some kind of cell cycle. Basically, it doesn't happen randomly. Everything is regulated, and there's usually some outside signal that's determining whether or not a gene is going to be expressed or not. Right? Makes sense? Of course. Okay. So this right here, let's turn the light off, is a microarray. Do people do microarrays anymore? I don't know. I feel like it's so 2000. Who does microarrays? I guess people do. It's just so much data. Every little spot there is a data point. What do you do with all that data? It's a lot. Anyways, So the point of putting this up, so first of all, let me explain what a microarray is for those who don't know. Who knows what a microarray is? Okay, good. So I see some hands, but it warrants an explanation. Don't go to sleep, any of you. No one. If anyone's sleeping, I'm going to throw things at you. I used to fall asleep if the professor turned off the lights. So, okay. So basically what you have on this chip is a whole lot of cDNA for each gene. So every single gene is represented on this chip. It's literally like a little glass slide like you put under a microscope. That's what it looks like, kind of, a little bit bigger. And a robot literally spots oligos in each spot. And then you do your experiment where you've got your control and you've got your experimental whatever. Or here what they're doing is looking in different tissues. So then they take those tissues, so they've got prostate, lung, brain, a whole bunch of stuff, and they isolate all of the RNA, right? So RNA that represents genes that are actively being expressed. And they make cDNA because RNA is not stable. cDNA is much more stable. And then they incubate it with this chip. And they, if it binds, if you actually have binding between your RNA and the chip because the RNA is there, then you're going to get green. And if you don't have it, you're going to get red. There's pseudo-coloring that happens later. Black means no change. So basically what ends up happening is you see up and down regulation of different genes. And so the point of this picture is just to show you that gene expression is cell type specific, which makes sense. But you can do this with anything. You can do this if you're looking at cells infected with a virus and not infected with a virus. You can do it in, you know, cells that are treated with a hormone and cells that are not treated with a hormone. Whatever you want to do, you just need that negative control. But here they're using it to show that different cells, so this whole line here, so what they're doing is they're showing the same genes going across, and then each way down you have different tissues. And you can see some genes, so like genes, there's a whole bunch of genes right here that in prostate are really low expressed, really high expressed, and in leukemia cells, are really low. So just want you guys to appreciate the fact that gene expression is determined by the environment. Every cell is a different environment. It's very fluid. It's not just one thing. Okay? Do you guys understand? You know what I'm kind of rambling on. No? Yeah? Okay. Okay. So what is she talking about? Gene expression is cell type specific. Okay. Different cells. So what follows is like a psycho over there. There's no shower. No. Sorry, guys. That's horrible. It's distracting. Okay. So what follows, if gene expression differs in different cell types, then clearly proteins will differ in different cell types, right? Because RNA translated protein, right? So what are we looking at here, okay? We're looking at a 2D gel, a 2D SDS page, 2 dimensions. Has anyone ever seen something like this before? A lot of like blank stares today. Oh, good. Someone waved at me. Good. Okay. Okay, so what is this? So basically, a 2D gel separates proteins based on size, right? So SDS page based on size. But then separates also based on isoelectric point. So your more acidic proteins are going to be at this end, and your more basic proteins are going to be at this end. So it's just a greater degree of separation. And the reason that was done in this example is just to show you how proteins are differentially expressed in different cells. So they're comparing here brain cells and liver. So this is actually like organs more than anything, because is there one kind of cell in the brain? No. But so in the brain and then in the liver, and you could see that it's actually really, these are really different blots, right? Would you say they're the same or different? Is this blot the same as this one? No, thank you guys. Thank you. Thank you. And there, so like an example here is there's this big bunch of blue right here up in the corner. Should I turn the light off? Let's turn the light off so I can get that one blinking again. Okay. So like right over here, there's this big bunch of blue in the corner, and it's not over here, right? Okay. So the way it works is you put all your protein on top, on the top, and first you run your isoelectric point, and that, based on charge, is going to migrate the protein left and right. And then you put it on top of the gel and run it down, and then it does it based on size. So this bunch up here is a really, really large basic protein that varies a little bit in charge, okay? And it's not in the brain, right? Not at all. I see nothing here, right? So you can think of them as kind of like copies of the same stuff, right? So another example is down here. There's a whole bunch of stuff that's not really over here. So different proteins in different cells. That follows, right? Different genes are expressed in different cells. You're going to have different proteins in different cells. And so, but wait a minute. What is she talking about? So do we all have the same DNA in all of our cells? Well, not all of us. Like, I don't have the same DNA as Kate here, right? We look a lot more alike, probably, right? No, but what I'm saying is within your own body, do you have the same DNA in all of your cells? Yeah, so what accounts for this difference? Why is this different? Gene expression is regulated differently. Yep, that's one thing. And what else is happening? There's a lot going on here, right? What can account for that? What seemingly is the same protein, right? Because this bunch up here, it's the same size. It just differs in charge. What might change the charge of a protein? pH. Well, let's say you have all of your proteins are at the same pH, but they migrate differently because their charge is different. So what's going to change? What can be done to a protein that's going to change its charge? Come on, biochemists. What can be added to a protein that might change its charge? I know, I'm being so vague, huh? Has anyone ever heard of phosphorylation? That's one thing, right? What's a phosphate group? Does it have a charge to it? A little bit? So that's going to change it. Okay, think about these things. Anyways, let's keep going. You'll start to see where I'm getting to. Yes?
Why are some dots red and some blue?
That has to do with comparing genes. I forget which is which in this picture, but I believe the ones that are I don't know in this picture, but usually when you do this kind of experiment where you're comparing two different conditions, you'll have one be one color, one be another color, and an overlap, so you can start to identify what's the same and different, but they're not using it for this purpose here. I'm not exactly sure why they've colored it in this picture. But it's straight out of the textbook and easy to look up. So we can look that up. Okay, so the next one. Gene expression. Okay, so what kinds of genes would you expect to be differentially expressed in different cell types? Come on, guys. Okay, the way to approach a question like this would be thinking, okay, different cell types. Well, let me think in my head of some examples of different cell types. Someone shout out a cell type.
Stem cells.
Stem cell, okay. Stem cells. Huh? And skin, okay, stem cells and skin cells. Would they have genes expressed differently? Oh, yeah, right? Insanely different, right? So what kinds of genes might be expressed in stem cells that wouldn't be expressed in skin cells? So you need to think to yourself. Yeah, go ahead.
Telomerase.
Telomerase. Yep. Okay. Telomerase. What does telomerase do?
It extends telomeres.
It extends your telomeres, right? So cells that are quickly dividing, cells that need to be stable. Okay. Telomerase is one. I was thinking of a very broad. That's a good one. That I'm thinking of a very broad class of genes that are involved. What's different about stem cells what's different from stem cells and skin between stem cells and skin cells?
They aren't differentiated.
That rapidly differentiating, is that what you're saying?
I said they aren't differentiated.
They aren't differentiating. Okay, so stem cells are pluripotent, would you say? They can do anything, so they might have a lot of things on, perhaps, that are turned off once they differentiate into skin cells. What do you guys think? Do you think I'm pulling this out of my armpit? Yes, so I have a it makes sense. What is she talking about? Okay. I just really want everyone to think about like things in a very holistic way in terms of just everything's part of a system. Everything's responding to something. It's not just like these finite things happening, because as we go into this, you're going to have to start thinking about large processes rather than just little details. So I'm trying to expand your minds a little bit. Okay, so let's talk about so my favorite is always if you're not pregnant, why would you need to make a placenta, right? Like if just thinking on a very large scale, right? So it all has to do, and so obviously there are genes involved that are turned on in some cases, not in others. Okay, so which genes would you expect to be expressed uniformly across all normal cells? And I say normal cells. What kind of genes? Yeah. You probably always need ribosomes. Yep. Housekeeping genes. So what kind? So exactly, housekeeping genes are going to be expressed uniformly. Who can give me an example? Ribosomal proteins was one. Yep. Other housekeeping genes. Yeah.
Genes for structural elements like the cell wall.
Yep. Genes for structural elements such as what did I have? What did we just blast? Actin. So actin, beta-actin, tubulin, those are very common housekeeping genes that tend to be expressed across all. Actually, if you look at gene expression experimentally, you usually need some genes to normalize by and you choose these housekeeping genes, and they're frequently tubulin, actin, ribosomal protein, beta microglobulin, which is part of MHC class 1 for those taking immunology. You don't need to know all this. We're just talking. We're just hanging out. Who brought some coffee? Someone has coffee. Okay, good. Actually, I was just remarking to my husband the other day that when I taught mole bio, it was 9 in the morning, and I always had a coffee. I know, I already drank my coffee. And I feel naked, actually, not holding my cup of coffee. So, anyways, okay. So how would so you've got all these housekeeping genes that are expressed uniformly across cells, you've got these differences. How would gene expression differ in cancer cells? What do you think might be expressed higher or expressed lower? Yes?
Well, they lack certain tumor suppressor genes.
They lack certain tumor suppressor genes, so like P53 is one, yeah. So what else? What do you think? What else do you think is maybe going to be expressed really low? It could be a class. It doesn't have to be a certain gene. It could be a class of genes expressed really low in tumor cells that are really helpful for cancer. Yes?
Probably anything that is going to induce apoptosis.
Right. Apoptosis-related genes, right? Because usually when cell cycle is disrupted, you go and you kill yourself, because you don't want to become a tumor cell. So a good thing that a cancer cell needs to do is shut down apoptosis. You got another one?
Anything that involves proofreading, probably.
Proofreading. Okay, so DNA repair. Yep. So frequently genes involved in DNA repair become dysregulated. Yep. So basically, you kind of end up in this kind of very error-prone state where your genome is completely unstable because you can't repair your genes, and you're having all these mutations, and you can't die, and you can't stop growing, and oops, you're cancer, right? Okay. So how would gene expression differ in infected cells? Let's say you're infected by a virus or bacteria is hanging out. What kind of genes might be increased, upregulated? Immune response. Yep. So like DNA replication genes for virally infected cells? Maybe. Actually, viruses are really good at manipulating gene expression and using host genes to accomplish their own function. So they might need to upregulate certain replication genes because they need to copy their own genome. Did I hear something from this corner? Did you say something?
Oh, I was kind of said what you said.
Okay. So yeah, so what happens if you're in uninfected cells and genes associated with the immune response are high? Autoimmune disease, right? Or allergy sometimes, right? Anaphylactic shock, that's like this insane activation. That's more activation, so I take that back. But autoimmune disease. Okay, so does anyone have any questions about what I'm talking about? Are you guys wondering why you're here? Who's wishing that they waited till next semester to take this with Dr. Snyder? Okay, good. Who's wishing they took this last semester with Dr. Snyder? Okay, just making sure. Okay, so here's a few things to think about. So there are 10,000, 20,000 of the cells through 30,000 genes that are expressed at any given moment, right? So, you know, 1/3 to 2/3 of your genome is being expressed. But the ultimate picture of protein expression, so that's at the gene level, right, and then you get to protein, it's way higher, right? So for every one gene that's expressed, there might be a lot more proteins made, right? So why would this be? Why would you get more proteins from one gene? What could happen that would give you more protein from one gene? Yeah. Splicing, alternative splicing, right? So we're talking about here post-transcriptional regulation. And then also, on top of that, you can have many functions of one protein, right? So you could have one gene result in 5 different proteins, which then could be phosphorylated, sumoylated, netylated, ubiquitinated, whatever, post-translationally modified, to go to different places and be turned on and be turned off and bind different things and function in many different ways. So actually, if you think of your genome's huge. We have a huge genome, sure, why not? Not that huge compared to some other organisms, but it's big, sure. But if you think about it as a functional genome, or your proteome, even more, your functional proteome, it's huge, right? So you need to think about things like post-transcriptional regulation and how post-translational modifications affect protein function. Yes?
Can you give an example of a protein that would be modified in 2 separate ways for 2 separate functions?
Oh my God, a million. P53. P53, the tumor suppressor, is netylated, ubiquitinated, phosphorylated. Oh my God, so much. Viral proteins tend to be insanely modified. I work on one gene, a herpes virus gene, which, if you look at its amino acid, it has something like 600 amino acids in it. So an easy way to approximate protein size is just move the decimal point. So you would expect a 60 kilodalton protein based on 600 amino acids. But it runs at like 100 plus because it's so highly modified. Because one phosphate group is only going to like bump it up a tad, but if you've got phosphorylation at multiple places, ubiquitination at multiple places, you get like a much larger protein. But P53 is a common one, and I think I have a figure. I don't know if I kept it or not because I think it freaked everyone out. But I have this figure somewhere of, well, we'll see if it comes up, and if not, I'll bring it on Friday. And it shows P53 doing a million things, and it's regulated by a million different modifications. Those who had 409 with me may have remembered that we talked about P53 and how it was modified in different places and did different things. But yeah, very common. That's huge. I work on post-translational modification, so I think it's like the key to everything. Okay, and you guys do too if you want to pass this class, right? Okay. So cellular processes can be regulated at any point from the initiation of transcription all the way to protein function. That's what we've been talking about this whole time, right? So you've got your DNA all the way to your active protein. You can have transcriptional control, right, whether or not you're even going to be transcribed. You can have RNA processing control. Are you going to be capped? Are you going to be polyadenylated? Are you going to be spliced, or how are you going to be spliced? You know, that all determines whether you're even going to ever make it out of the nucleus. And then you have RNA transport and localization. So RNA needs to get to the right place so it can be translated and then be in the right place. And then, is it even going to be translated? Is the RNA going to be degraded? That's a really common way of regulating things. And, you know, if it's degraded, then, well, there goes the end of everything. But if you're translated, then you got protein, and then your protein activity can be regulated based on how you're modified and where you go. So think of it in this big picture kind of way, right? So transcriptional control, RNA processing control. What do I mean? So then there's post-transcriptional control, right? So we kind of talked about that for a second there, right? You can have attenuation, right? So that's where you don't get complete transcription. You can have RNA editing. So that's where you're actually the RNA is going to be edited to either maybe insert a stop codon somewhere or change something somewhere, and that's very common in, I think, Drosophila, and in a lipoprotein. Which lipoprotein is it? I forget. Then nuclear export. If any of these things don't work, you're not going to ever get out of the nucleus and you're going to be degraded. Why would you want that to happen? It's kind of quality control in a way, right? Localization, translation, we'll keep going. So RNA destabilization. Okay, so how are we doing on time? Okay, so you want to go through this? Okay, let's stop for right now, and we're going to quickly look and see about finding the textbook online before I forget that.